Have you ever noticed a fence line completely obscured by piles of plant debris? Had a tumbling weed smash into the front of your car? Perhaps you've seen a conspicuous line of seedlings appear across a plowed field. Today we are going to be discussing the invasive weeds diffuse and spotted knapweed. I'm Christy Gladham. I've worked in biological control for over 10 years. Welcome to the Colorado Department of Agriculture's Biological Control Series in collaboration with the USDA Western Sustainable Agricultural Research and Education Program. These two plants, Centaurea diffusa and Centaurea stoebe, are weedy rangeland species introduced from Europe in the late 1800s as a contaminant of alfalfa seed. They are not the only invasive knapweeds around. Russian knapweed is also very common in the western U.S. A positive identification of the weed species is necessary for proper management. So how can we identify the knapweed species? There's two easy ways: by their roots or by the leaf-like structures that cover the bottom of the flower heads called bracts. Diffuse or spotted knapweed have a singular large taproot that is creamy white. Russian knapweed has black, scaly, spreading roots. For the flower head bracts, Russian knapweed has papery bracts, while diffuse or spotted knapweed have comb-like bracts with 4 to 5 lateral spines. Typically, spotted knapweed is differentiated from diffuse knapweed by the flowers. Spotted knapweed has purple flowers and dark terminal ends to the bracts on the flower heads, which give them a spotted appearance. Diffuse knapweed typically has longer, more distinct terminal spines at the tip of the bracts. If these are hard to see in the field, a jeweler's loupe could be used, or you might open a photo app on a cell phone and zoom in. Utilizing the bracts to differentiate diffuse and spotted knapweed won't always work. These two plants can hybridize and form intermediate varieties that have a range of physical characteristics that don't quite fit into a single species category. Luckily, the growth, control, and management of diffuse and spotted knapweed takes the same approach. For this video, I will refer to both species as just knapweed. Knapweed propagates only from seed. The seeds may germinate at any time of the growing season when enough soil moisture is available, and while in the soil, the seeds are thought to remain viable for 5 to 10 years. Once the seeds germinate, the plants form a rosette of leaves close to the soil surface, which will grow or overwinter until the rosette reaches a critical size. This may take less than a season or several years. Once that critical size is reached, the plant will begin the flowering process by sending up a central stalk. This is called bolting. The bolting plant will reach 1 to 3 feet in height and will be shaped either a bit like a Christmas tree or like a ball with many fairly stiff branches. The knapweed forms singular flowers near the tip of each branch. These flowers are urn-shaped, white to purple in color, and about 1/2 inch across, or about the size of a nickel. Each flower will produce many seeds. In total, a single plant produces around 18,000 seeds, with up to 40,000 seeds produced per square meter of infestation. Once the seeds mature, the plant dies and dries out. The plants can break off at the soil surface, and the bushy growth allows these plants to roll across the landscape, widely distributing seeds as they are windblown or carried along by vehicles and equipment. This is why you'll often see infestations where these tumbleweeds either get caught or in the open areas that are windblown, so most commonly along fence lines and roadways, but always in areas that might be considered degraded. Besides making a mess of fences and dispersing such long distances, knapweeds are also thought to unfairly outcompete native vegetation. For a while, this is thought to be through allelopathic chemicals. These are compounds exuded through the root system that actively suppress growth of other plants. Further research has shown knapweed can form relationships with soil fungi that steal nutrients from nearby native plants and give the nutrients to the knapweed. This greatly increases knapweed's competitive ability while actively harming native plants. Knapweed can also decrease forage since it is quite bitter. Most animals will avoid grazing on it. Yields might also be decreased because of contamination of agricultural products. How can we manage knapweed? The large taproot of diffuse or spotted knapweed can be severed or pulled to kill the individual plant. These taproots may reach over 2 feet deep in as little as 40 growing days. Mechanical removal can temporarily cause a higher population of knapweed, since the disturbance can allow buried knapweed seeds to reach the soil surface and germinate. If the plants can be destroyed before they flower and set seed, then the infestation can be managed. Mowing is not typically beneficial, as it can both spread seeds from already flowering plants and also cause the plants to just flower lower to the ground. Herbicide can be used on rosettes at any time of the year, but can also create patches of bare ground where invasive weeds are the most likely to grow. Mowing could be combined with herbicide treatments to reduce non-target plant coverage and improve herbicide application directly to knapweed. Management goals can vary widely. If eradication of small, discrete patches of knapweed is desired, biological control will not be the right strategy to employ. In those cases, herbicide application or hand-pulling rosettes could eradicate patches within a couple of seasons. However, if you have large, extensive patches of knapweed, and the goal is to reduce the density of knapweed on a longer time scale of, say, 5 years, reduce invasion potential for neighboring properties, and reduce disturbance of the land as much as possible, then biological control will be the right choice. As always, before considering collecting biological control agents, please ensure you have the proper permission or permitting to do so, and ensure that biological controls are permitted and allowed in your areas. Movement of biological controls across state lines is permitted by the USDA APHIS PPQ and subject to rules and regulations. Around a dozen biological control agents have been released for control of diffuse or spotted knapweed. There are 2 successful and well-established biological controls that I will highlight. Knapweed Seedhead Weevils, Larinus obtusus or Larinus minutus, have been in Colorado since the early 1990s. They could possibly be variants of the same species, though further research is needed. It would be extremely difficult, if not impossible, to differentiate them in the field because their life cycle and habits are nearly identical and their physical appearance is incredibly similar. However, several field researchers have described habitat and plant preference differences between the two, so if redistributing these weevils, it is a good idea to match the release site with conditions from the collection site. These would be conditions like rainfall, plant community, temperature range, or dominant knapweed species. Knapweed seedhead weevils of either species are around 6 millimeters long, or about the size of a black-eyed pea. They're blackish gray with a very stout snout. They overwinter in the soil as adults. They emerge in early summer to feed on the knapweed plants and flowers before laying eggs. The females have to feed on knapweed flower heads for their ovaries to develop, so egg laying only occurs after knapweed has begun to bloom. Up to 5 eggs are laid directly in an individual seed head. When the eggs hatch, the larva will feed within the seed head and destroy 99% of seeds. While still inside the seed head, the larva makes small chambers out of chewed seeds and pappus hairs, where they will pupate or turn into adults. Once fully developed, the new adults will chew their way out of the pupation chambers, which leaves a very characteristic hole and empty chamber in the seed head. These holes in the seed heads are a great indication of seed head weevil presence and can most easily be found in the previous year's stems. When these new adults first emerge, they may be covered in slightly orange-colored fine hairs. This can help you differentiate between the overwintering and new adults. Once the new adults have emerged, they will feed on the knapweed plants and then drop into the soil to overwinter and start the cycle again the following year. They have only 1 generation per year. Collecting seedhead weevils is straightforward. I like to scout sites in early spring and check the previous year's flower heads for those telltale emergence holes. I check the ground for knapweed rosettes to make sure there will be enough plants to collect from, and will plan on visiting good sites for collection in late June to early July. The best timing is approximately 1 week after the knapweed plants are in full flower, and collection can continue for several weeks. I don't typically collect in August in Colorado, but weather conditions can shift the season of collection by a couple weeks in any year. Any day chosen for collection should be rain-free on dry foliage. If insect nets get wet, it greatly increases the chances of damaging or killing the collected insects. Sunny days are usually better, but the hotter it is, the more the insects may want to disperse. For collection, you will need an insect net with heavy canvas, not gauze or netting, gloves, long sleeves, insect collection container with lid containing some knapweed foliage, a cooler to store the collected insects, and optionally a sorting tray with forceps or aspirator for removing bycatch depending on the release site. I strongly recommend the gloves and long sleeves, as knapweed can be quite irritating and noxious. Make sure to wash your hands before eating or touching your face. It usually only results in an extremely bitter taste, but some people could have worse reactions like severe rash. For the insect collection container, you could use cardboard containers, a pillowcase with a drawstring, or a plastic container that has a screened hole in the lid for ventilation. To collect the insects, hold the insect net with the hoop 45 degrees to nearly flat below the bulk of a knapweed plant. Use your other arm and hand to gather and tap the knapweed across the opening of the net 5 to 10 times to get weevils to fall into the net. You can make your way across the knapweed infestation and periodically shake down the net to concentrate the insects at the bottom. When the net has a few hundred weevils, you can empty it into the insect collection container. Make sure to put the lid on well and keep the container either in the shade or in a cooler with ice packs so you don't overheat and kill the collected weevils. It is possible to go over the same knapweed a couple of times in the field with at least 10 minutes between tapping. A good set of taps on a plant should yield at least 10 adult weevils, and sometimes up to 40 or 50. If you are getting less weevils per plant, then a couple of things could be happening. Either the weevils present have not built up a high enough population to be collectible, the timing of collection is too early or too late in the season, or if you can see lots of weevils on the plants, you may not be tapping the plants hard enough. Once insects are collected, either you've gone over the entire patch or you have enough insects in your collection container, it's time to make a decision on whether to sort the insects or not. If the site you have collected from does not have other noxious weeds, so you're not worried about moving seeds and risking different infestations, also your release site is very nearby with a similar ecotype, you might forego sorting the insects and just release the bulk container that you have just collected. However, if you are concerned about introducing other weed seeds, pest insects, or other contaminants from the collection site, or maybe you want to release in several different sites, then I would recommend sorting insects. For this, you will need a sorting tray, an aspirator or forceps, containers for the sorted insects, and a cooler or refrigerator. The containers should be roughly pint-sized and contain non-flowering knapweed leaves and stems as food. A dry paper towel can also sometimes be placed in the container to help control humidity levels and provide cushioning if the insects need to travel a long distance. To begin, you will want to cool the collected weevils. They may already be chilled out enough from placing them in the cooler in the field. Cooling them helps to slow down their movement and prevent them from flying while you sort them. Make sure you do not freeze them, but any refrigerator around 40 degrees Fahrenheit will work fine. Place weevils into a sorting tray or container. We use photography chemical trays, but a baking sheet or other rimmed flat container will work just fine. To sort the insects, you may either use forceps or an aspirator. If you use forceps, you may place seedhead weevils individually into a release container, and if using an aspirator, they will collect in the aspirator's collection tube. Count as you go and aim for 200 insects per release cup. This will be enough insects to begin a new infestation. I normally recommend around 1 release per acre of knapweed infestation, but a higher release rate will just establish insects a bit quicker and provide faster control. Releasing fewer insects may cause the weevils to not establish or to take quite a bit longer to build their population. The second biocontrol agent, Cyphocleonus achates, the knapweed root weevil, is a very large, mostly flightless weevil. They are around 20 millimeters long or the size of a peanut, with mottled gray to brown coloring. The adults have around a 10-week lifespan from late July to early October. They spend most of their time just at or below the soil surface near the root crown of knapweed. The adults will climb to the top of plants on sunny, warm days to find mates. On the plants, they could almost be mistaken for an unopened flower head because of their size and spotty coloration. After mating, the root weevils will lay their eggs just below the soil surface on the root crowns. Once the eggs hatch, the larvae burrow through the taproot and will overwinter there in their larval stage. Pupation occurs the following summer, and the adults emerge to complete their 10-week lifespan. These weevils typically only travel along the ground, so roads or trails can be substantial barriers to their movement. Try to release these insects in areas where there is a continuous corridor of knapweed on the ground. To survey for establishment, root samples may be taken and dissected to search for larvae from October to early June. However, root sampling is destructive and will kill any insects found. There are also several other root-feeding species that have been introduced for control of knapweed, so it is important to identify larvae correctly. To collect Cyphocleonus, you will need containers for the insects with knapweed foliage, a click counter, and a cooler with ice packs for transport or storage. The adults may be readily found on the very tops of plants, typically in August. They can be startled and will drop off the plant and play dead for up to several minutes. With their coloration, they can be extremely tricky to spot once on the soil surface. It takes a little while to develop an eye for cyphos on plants, but once you begin to see them, they become more and more conspicuous. They could be collected with the same tapping method as the seedhead weevils, but I have found that I get very poor yield of collected weevils from the total number of weevils that I visually spot. For these reasons, I prefer to hand collect those weevils. Once you have spotted a weevil, you will want to approach the plant. Carefully place one hand under the weevil. Using your other hand, you may try to grab the weevil or knock it off into your waiting hand. The weevil may then be placed in the collection container. These weevils can be much more solitary than the seedhead weevils, so a typical release is 50 adult individuals. Again, releasing more individuals might provide faster establishment or better control sooner. These little weevils can be very strong and may cling to your hands. Sometimes blowing gently on them can get them to relax their grips, or a small piece of foliage can be used to brush them off. Be careful not to damage them. If you are hand collecting cyphos, they will be sorted directly in the field and ready for release in other infestations or shipment. Releasing either the seedhead weevils or root weevils is incredibly straightforward. Prepared releases in cups can be stored for up to 1 week in a refrigerator, but releasing as close to the collection time is better for the health of the insects. Take your release cups into the patch of knapweed you would like to introduce weevils. Usually mid-morning on a sunny day is the best time for release, as the insects will be more active, but any time of the day should be okay. Don't release in very heavy rain. If it rains after release, it is okay, the insects will be fine. Open up the container and tip the weevils out into the knapweed all in one spot. They will disperse themselves throughout the patch, so you should not scatter them. If they are clinging inside the cup, you can take a bit of grass or soft foliage and gently brush them out of the cup. Weevils have a startle response, which can cause them to play dead. It can be very convincing, but they should begin moving about in a couple of minutes. I like to take a photo of the weed patch for future reference, a GPS point, the number and species of weevil released, and the date of release for my records. You should begin to see the results of your release within 2 to 3 years. It takes time for weevils to establish their populations. Adult seedhead weevils at high population levels can have serious defoliating effects on knapweed. This could possibly be seen within your infestation a year after release. The knapweed plants under the most severe attack may change color from a light green to a bluish green, with distorted growth and fewer leaves. The pupation chambers, and most especially empty pupation chambers, should be readily seen within the dried seed heads. While the seedhead weevils will prevent most new seeds from being produced by knapweed, they do not prevent new plants from coming up from the seeds already present in the soil. The root weevils may outright kill small rosettes through larval feeding, or the plants may be stunted. It's also possible for larval feeding and tunneling to open the plant up to secondary infections from soil microbes, which can be lethal. In some areas, seedhead weevils may work better, and in others, root weevils might work better. It just depends on the establishment and site conditions. Utilizing both seedhead and root weevils together may provide better control of knapweed than either alone. The seeds contained within the soil below an infestation can lay dormant for several years, though exact numbers are not known. It can take time for the soil seed bank to be depleted, and during this time, new plants can germinate and grow. The seedhead weevils established will help to prevent those plants from producing as many seeds, and the root weevils may kill some rosettes, but neither will fully eradicate knapweed. Most other control methods will not be compatible with either the seedhead or root weevils. It is especially important to not destroy the developing larvae of the Cyphocleonus within the rosettes or the Larinus larvae in the seed heads. However, the weevils may reduce knapweed populations to a point where other control methodologies will be more feasible in terms of time, input, or cost. These controls can be used to eradicate small infestations over a couple of years, and the weevils will continue to provide landscape-level suppression of the knapweed. A healthy landscape with lots of competitive native vegetation may see better and faster results from biological control. Strategies could include tillage-free reseeding and providing supplementary moisture during the growth stage of desirable plants. I hope this video has given you the information, methods, and confidence to identify, collect, release, and manage knapweed seedhead and root weevils. There will be a list of references and sources in the video description if you would like to find out more about these helpful insects and how you can manage knapweed. Thank you for joining me today.